Okay, we're going to have kind of an open session for industry, and these are we've got up here 5 terrific industry members who have either ALD therapy currently in trials, recently in trials, or in early development. And so we're going to give an opportunity for each of them to introduce themselves, spend a few minutes telling you about what they have in the works. And I think the goals of this session are really to allow you to hear what the future holds, at least in terms of potential trials. Clinical trials for every community represent a hope for new therapy. This is really kind of the culmination of a lot of what we're doing here, is trying to get to this point. And these representatives are from companies that are trying to help deliver those therapeutic options. So it's really a pleasure to introduce the group here. I'll let them introduce themselves, but thank you all for being here.
My name is Mandy Cromwell. I'm from Genzyme Sanofi, or Sanofi Genzyme. Some of you in the audience will be familiar with Genzyme as having been I guess a pioneer in rare disease treatments, and we are doing a research initiative in AMN, ALD. But I have to say, as the first person, we don't have a clinical trial and we don't have a drug in development yet, but we do have a research program for AMN. Our approach is, I think, different from the ones that you've been hearing about today. It's substrate reduction therapy. This is something that Genzyme has had some experience with and some success with in other genetic diseases. Notably for Gaucher disease, we have a drug called Serdelga. And so we've expanded our approach to some other genetic diseases, including AMN. And we think this would be, well, first of all, this would be a small molecule program. So it's an oral drug, potentially a pill, something that could be taken throughout life. It's potentially therapeutic for all of the disease manifestations, including AMN and CALD. And we think it could also be complementary to the other types of treatments, including stem cell transplantation and gene therapy, perhaps supporting those approaches in a combination therapy during engraftment. And so I think those are all the points I wanted to make about that.
Maybe what we'll do is we can save questions to the end, let each of the speakers present their industry's molecule or program, okay?
It's number 2. Yeah, you can hear me. I'm Uwe Meitsch, Chief Medical Officer of Minoryx. As you heard from Florian this morning, our trial has just closed enrollment. So we have reached our enrollment target, which is on one hand good news. Actually, we are 2 months ahead of schedule with recruitment. That means we get the results 2 months faster. It may have a disappointing side for patients who try to get into the trial and now hear, sorry, we're not scheduling any further appointments because the trial is fully enrolled. It sounds a little bit harsh, but on the other hand, before we start a trial, we have to commit to regulators to enroll up to a certain number of patients. We are not allowed to over-enroll, and if we did that, we would have to ask for a substantial amendment, which would have to go through regulatory review, which takes 1 or 2 months, which would just extend the trial. So that is the reason why we closed the trial now for enrollment. As you all know, it is a 2 years double-blind placebo-controlled study. That means in about 2 years from now, we can close the database and we will have results, and we all hope that we will have an approvable treatment available by that time. What is now key, I think, is for all the patients who are in the trial to manage expectations. So one thing is Min-102 is not a cure to the disease. It is a compound that, from all that we know, is able to control the degeneration, the degenerative processes. That means it will be able to slow down, or in the very best case, to stop progression of the disease. Expectations of improvement may be very nice to have, but I think are almost too good to believe. So to all patients, please be patient. Please comply with the study procedures. We know some visits are quite a burden to the patient. The reason is that we try to cast a very wide net of outcome parameters. When we planned the trial, there was really nothing out there that could tell us which is a sensitive parameter, which is indicating change, delay of progression within 2 years. And therefore, we chose a very wide variety of outcome measures, which are time-consuming to assess during a visit, but we think are probably necessary, because even if we fail the primary endpoint, regulators will look at favorable trends on other endpoints. So the more endpoints we have, the more chance we have to detect a signal. Another thing is every patient on the trial may really try to find out, am I on active or am I on placebo? If there has been a law like if you don't have adverse events, you're on placebo, I don't think that is true from all what we see. So when we look at incidence of adverse events, it is definitely lower than you would expect if all patients were on active treatment. So even if you have no adverse events, it may well mean you're on active treatment. Yeah, I think that's all that I have to say. So the final reward for everyone who stays on the trial over 2 years is that we offer an open label extension. We are now also offering a lot of assistance to patients who live far away from sites. In the United States, we offer that certain visits can be performed at the patient's home to avoid travel. We have certified nurses going to the patient's home and making the assessments. In Europe, we don't have that, but we have less frequent safety visits, and there we are also offering travel assistance like flights, hotels, if that is absolutely needed. So on our end, we hope we have done everything to make the patients' lives as easy as possible. But again, we have to count on patients' patience, and we hope also that the investigators who are here, and there are quite a few investigators in this room, try to keep the spirits up of their patients, to motivate them, to explain that it is time-consuming, and some visits are burdensome, but we hope it is worth the effort.
Thanks, Uwe. Hi, I'm Sheila DeWitt. I am the president and CEO of DoTareq. Recently, our lead program for AMN, DRX-065, was acquired by a French publicly traded company called Poxel. So you will see Poxel as a sponsor of this event. Poxel is focused on diseases of metabolic disorders. And so our program is aligned with their strategy. We are PXL 065, I need to change the first 3 numbers in my head, is also related to pioglitazone. You may have heard, as Florian discussed earlier this morning, that pioglitazone, as Aurora Pujol, who's also here, has studied in animal models, reverses the disease progression and is believed through mitochondrial and anti-inflammatory effects. The interesting thing about pioglitazone, you may recall, I've told this before, but it's a mixture of 2 compounds, and they chemically interconvert, and what we discovered is that one of those is responsible for the mitochondrial and anti-inflammatory activity. And so associated with the other activity with pioglitazone is a PPAR gamma effect, which can lead to weight gain, fluid retention, and risk of bone fractures. And so what we're trying to do with 065 is improve the therapeutic profile on pioglitazone. We are currently in phase 1 clinical trials. We will also be looking at another indication in full disclosure, which is non-alcoholic fatty liver disease, or NASH, which is also an area of very active interest. About 15 million Americans have it, about 50% of type 2 diabetic patients. So we continue to consider both indications, but at this time hope that the benefits we see in the NASH patient population may also transfer into the AMN patient population. So with that, that's the background.
Jeff Cleland, I'm the chairman of the Orpheris. We're a small startup company that spun out of Johns Hopkins University, and I think this is my third time. So in the last year, we've completely transformed the company into a neuroscience company, so I encourage everyone to look at the new website. We actually have 4 compounds with proof of concept clinical data in ALS, MS, Rett syndrome, and our lead compound is the one that we're taking forward into CCALD. This drug actually is relevant to the last speaker's talk because we've been able to show in 25 animal models that when we administer this drug in acute neuroinflammation, we can put the fire out. And that's really going to be important, we think, in CCALD. And then more recently, we've done work with Dr. Fatima and Dr. Turk, who's just published a paper on this, where we've been able to take patient samples from CCALD patients and show that actually we can return them back to normal with treatment with our product, our lead product, which is called OP101. And then we just finished a healthy volunteer study where we had no adverse events at very high doses IV, and we're going to transition over to subcutaneous dosing with this product. And then very lastly, I'll tell you that FDA has actually approved us to do a phase 1, 2, 3 trial, so a single seamless clinical trial all the way to approval. They've agreed on the statistical endpoint, the primary endpoint. And so Keith actually will be presenting some data on that tomorrow, slides from what we're going to be doing for the trial. So why aren't we doing the trial yet? Because we don't have money. So hopefully, hopefully we'll have funding soon. I can tell you good news next year that we've already started the trial. Thanks.
Great, and I'm Gary Fortin. I'm with Bluebird Bio, and we already heard a little bit this morning. Bluebird Bio is developing an autologous lentiviral-based gene therapy for cerebral adrenoleukodystrophy. We're excited that we've met our enrollment target, so we've enrolled all 30 patients in the trial, which we didn't plan it this way, but it did turn out to be a phase 1, 2, 3 study as well, and we anticipate that trial for registration. The first 17 subjects have completed the 2 years of follow-up, 88% alive and MFD-free at that 2 years. Some patients now out 5 years post-treatment. And as I mentioned this morning, we're anticipating filing with the FDA in 2019. So with that, I'll turn it over to Keith.
The green mic here. So maybe I'll start off here with a question for industry here, and maybe let everyone speak to how their company chooses, in some sense, tell people a little bit about how, and I'll let whoever wants to answer, how you choose your target form of ALD, which form of ALD you want to target, and maybe a little bit about how you design your trials. I think that's something that might be helpful to some patients and families, because many people out there may say, well, why don't we have a trial yet for the form of ALD that my son, daughter, or self have? And or why doesn't this trial include more patients so more risk be enrolled? Anyone want to speak to one or more aspects of that?
Yeah, I gave a talk exactly a year ago here in Philadelphia about how we chose a target. So we did a stepwise approach in non-clinical experiments from in vitro, so that is in the test tube, to in vivo, single and double ABCD1 knockout mice. We looked at biochemical readouts, we looked at behavioral readouts in the animals, and concluded that it is able to influence the degenerative cascade in various ways. We also have results from very classical neuroinflammation experiments. There's no specific paradigm for CILD, but there is a paradigm everyone knows for multiple sclerosis. It's the EAE model. It works also anti-inflammatory in that model. That means now it is probably amenable to CILD as well. Why did we not choose CILD? I always say in an ideal world we would target all indications at once. So all phenotypes. Now we chose AMN men as the first one because it's the most frequent phenotype. I've been asked women. That study is likely to follow if the first study is successful. We are also currently thinking of going into CALD, given the anti-inflammatory properties of the drug. But as I said, in an ideal world, we would do everything at once. We have limited funding, so we have to make conscious use of our funds. It is our principle that we do not start a trial if it is not fully funded to its planned end, and that is the reason why we don't pursue all 4 phenotypes at once. Yeah, maybe I stop here and talk about other things later.
So we actually looked at the population that we wanted to treat, and a lot of this came from our advisors, including Keith and Ali and Florian and Troy and others. And really, I think what we learned is that from that experience, and also talking to the FDA, was that we wanted to go after an unmet medical need, a clear unmet medical need, and unfortunately, Bluebird solved hopefully the first part of the problem. So we're looking at patients that are not eligible for bone marrow transplant as our first population, because they really have no options. And so by doing that, we think we have a better chance to move quickly through the regulatory process and also quickly show benefit to patients. And so we actually picked that pathway just because of that unmet medical need and the potential there to really show quickly in a very small number of patients the benefit of our drug through a reduction in gadolinium intensity score, hopefully a reduction by PET TSBO, which is what I asked a question about earlier today. But just really being able to show we can put the fire out in a really extreme case and then move to the other cases as we go forward. So that was our strategy, and that's the rationale for how we went forward.
Well, let me comment. As you think about pioglitazone, I don't know if you're aware it's a generic drug. It was first approved as Actos in 1999, so it's been around for 20 years now. But it's not approved in children. And so one of the reasons we started by looking at AMN as opposed to CALD is to first address the larger patient population, and there's really a lack of clinical trials in the AMN. I mean, Minoryx is really pioneering it here, and it's also related to pioglitazone. So for us, that was important. We did meet with the FDA when we first were filing to go into human clinical trials, and they were very intentional about us developing a plan for pediatrics as well, recognizing that that would be the next to come after the AMN.
So Bluebird Bio actually has Patrick O'Borg to thank for kind of our inspiration of why we went down this path of gene therapy. Back in 2009, I believe, Dr. O'Borg in Paris published proof of concept data in patients with cerebral adrenoleukodystrophy, lentiviral-based gene therapy that, although not the same as Lenti-D today, was kind of a precursor of Lenti-D. Treated 4 patients and with most of them doing very well post-treatment. So based on that, we partnered with him to develop Lenti-D and then move Lenti-D into the clinic and into registration trials. How we designed those trials is in partnership with FDA and EMA in Europe is we did a retrospective natural history study to really understand what is the typical disease course for patients with cerebral ALD, as well as what are outcomes that we can expect with allogeneic stem cell transplant. And based on that retrospective analysis, working with various investigators and experts in the field, aligned on this novel endpoint, recognizing that it's not enough for these boys to just survive, but it's important to survive functioning with the quality of life that they deserve. And with that, we designed an endpoint that was 24-month survival, MFD-free, or major functional disability-free survival. So it was really coming out of that natural history study that we understood the clinical relevance of that kind of endpoint. I guess that's it.
Walk into the audience here and see if there's anyone who would like to ask questions here for any member of the Industry Council or just in general.
Hi there. So, sort of pointing at you, sorry I've forgotten your name. I think it's great that you're making sure that you have adequate funding before you embark. As many of you in the room will be aware of the Neurovia trial, where we actually had in the UK a boy who was ready to start. So the phone call kind of went, you're being enrolled, but actually we can't start because funding has been lost. And of course now, as I understand it, that trial isn't going ahead. So I just think it's not so much a question, it's just that actually it's really important when I support these families and you're given a glimmer of hope and then suddenly it's snatched away from you. So I think it's just so important that when it starts, you see it through.
Yeah, thanks. I will say that for those of you who have been involved in any clinical trial of any kind as a patient or family member, you may get the sense that there are a lot of hoops to jump through, and as you probably can guess, many of those hoops are very expensive. Not all of that is by the choice of the sponsor. Some of that is actually required by the FDA for safety purposes or for other kinds of purposes. But the cost of trials is hugely expensive, and it is a limiting factor to launching and then eventually in the pricing of the drugs. It's a very hard industry to navigate as a funder and as a industry driver. Other questions? Is there one from Alex? Go ahead.
Alex Sherman, Mass General. So in other diseases and in general, there is a grassroots movement and the trend to ask for open access to Hello? Yeah. So open access to your drugs or your treatments even before they were approved, during your investigational phase. And I do know that there are a lot of pressures and there are also a lot of thoughts on behalf of the companies because it's financial liability, it's just financial burden and so on. So I was wondering whether you experienced such things here in ALD and how are you planning to respond or how are you responding? I know, so.
Your questions about like expanded access or open access in a development stage. So yeah, so to date we've limited access to LentiD in the context of a clinical trial, and that's the policy of the company pending a formal benefit risk assessment by regulatory agencies. However, at the time that we developed this policy, we anticipated enrollment in 102 to last a little bit longer than it did, right? So we had an enrollment target of 30. Based on earlier enrollment projections, we anticipated that it would take us until 2019 to enroll that population. We published interim results of the study in the New England Journal of Medicine last year, and enrollment rapidly ticked up. And so we ended up meeting that enrollment target a lot earlier than we thought. So we were in a situation where we had to anticipate, how to address patients, how to provide access to patients who needed it. And in consultation with investigators and with the community, decided that instead of an expanded access approach, we would actually open another clinical trial. And so in early 2019, we're actually going to initiate a novel clinical trial. We just announced this actually yesterday publicly, so you guys are first to know. And that trial will have the same eligibility requirements as ALD 102, however, is intended to assess a more contemporary conditioning regimen. So back 6 years ago when we started this trial, it was standard to use busulfan plus cyclophosphamide. Over the years, that's evolved a bit, and so in the new study, we'll be assessing busulfan plus fludarabine. And we will also not be limiting eligibility in this study to patients without a matched sibling donor. So at the discretion of the investigator and the family, even with a matched sibling donor, if they deem eligibility as appropriate, they'll be allowed in this study as well. And so that's how we are enabling access, but we're not going down the broader expanded access approach.
Yeah, we have also thought about it, and we are currently refraining from that. One reason is that regulators see it with a lot of suspicion, so as if we were trying to get approval through the back door if we collect further data. It is currently unproven whether the drug is efficacious or not, so we would expose many patients potentially without knowing whether the drug works or not.
So is it different in Europe?
No, we don't make a difference between Europe and the US, so that is the main reason why we are not currently considering compassionate use.
I have probably a more higher level question that you guys may or may not feel like you could address, and it's with regards to clinical trials, something I've thought a lot about. As an adult man with AMN, I have learned that the FDA is not necessarily the greatest institution on the earth when it comes to moving drugs through trials and efficacy and figuring out if things work. The FDA is kind of hyper concerned with safety, as they should be. I don't really care that much about safety. I would chop my left arm off to fix my AMN. Safety's kind of like, I'm a guinea pig. I'll try anything out. So the question is, why, on a higher level, why don't we have something in the United States where I could just sign a form waiving my rights, saying no liability on you guys, send me that drug. Let me try it out. I just sign away. You just send me whatever you want. We'll just, it won't be an official FDA approved clinical trial, but I don't care much about that. I just want to try out what you guys are doing because it sounds really awesome and exciting. And if I wait on the FDA, it's going to take 5 years, 10 years. And I don't want, it's like, let's go. So I'm wondering if any of you can address that question of why can't I just forego that with a signature and say, send me whatever you got, I'll do it. If you need to send me into outer space, I'll get on a ship. I'll do whatever you guys want. Like I said, I'll chop my left arm off. I'll do all kinds of crazy things.
So I don't know if you're familiar with the right to try legislation that was passed not that long ago. So but it has to be life-threatening. So I don't think necessarily, I hope you don't qualify. So there is that, but I think that it's back to what Gary said earlier, is that every company has to look at the risk-benefit. So we appreciate and understand why you would want to do that, but at the same time, if you have an adverse event or something that we wouldn't have expected in another patient population, we have to report that. So then it puts our drug at risk for maybe getting approval for what you're ultimately hoping to see, which is a drug approval in your indication. And so it does put the drug, not necessarily the company, at risk in going into populations that are not well characterized, that we don't necessarily understand as a company yet how the drug's going to work. So that's the trade-off.
I understand the logic of the FDA process. And I understand why it exists, why we have it, time frames, and all the steps, the procedures, and the hoops you have to jump through.
But the FDA doesn't have AMN, and they don't care about that. They're concerned about, the general United States population and all this kind of stuff. So when you're in my kind of predicament, I want to travel to China. I want to go anywhere. I want to try out anything and see if anything can help, even a little bit of an improvement. It's like, we live in the year 2018. There's so much technology. There's so much out there. You guys are all working on these amazing therapies. And I just think, well, I'm just going to have to duke it out and wait around.
The story of how we ended up where we are with the FDA. How many of you remember thalidomide from the 1960s in the story? That's why Helen Kelsey, who refused to get it approved in the United States. So thalidomide was approved in Germany and many other countries in the 60s for nausea with pregnancy. And there was a company, pushed and pushed and pushed and pushed for it to get approved in the US. There was some data that was not fully disclosed to the FDA at the time, and it was never approved at the FDA. But in the meantime, a whole series of birth defects happened throughout the world, and the US was essentially spared because of this woman, Helen Kelsey, at the FDA. And so that wasn't that long ago. It was in the 60s. So that's the reason. I mean, we think our drugs, our drug candidates, they're not drugs yet, are safe, but there's a chance that they aren't. So I want to just put that out there.
I mean, I think we know that the Minoryx drug is, for the most part, pretty darn safe, because I don't think anyone's died from trying your drug yet. I think we would all know that if that's happened. To a greater or lesser degree, people are experiencing some things. You know, we still have a long ways to go. I get that. So it's just, I'm just thinking pie in the sky about the state being a diseased person, yet having to play by the rules. You know?
Justin, I want to give the floor to some other people too. We can come back because I mean, the larger point is a really important one of, why don't we have more autonomy to do what we want, do what we feel they really need to do, and be able to assess our own risks. And it's a very large question, and but I think all of us can appreciate the sentiments behind that one. Thank you. I think Ben's got a question here too. I want to make sure he gets.
Yeah, a few improvements that industry could make alongside regulators. So I think one is to get a report on failed or inconclusive clinical studies. You know, that is still not happening. So one example is a few years ago there was a big study in Europe that was backed by ELA for high-dose biotin with a company called Mandy. That trial came to an end, and there was no lessons to be learned from what worked, what didn't work, what they learned. So that's I think that would be a great policy to have, to really dig into clinical trials, if they succeed or if they fail. The other thing that's been a little bit the bugbear of this landscape is biotech companies that are very hot in our community. Our gold sponsors of this event say that, make public pronouncements, they're fully committed to our population, and then go dark. And that's happened recently with one that was a spin-off of Novartis in San Francisco. Totally went dark after making big pronouncements in the ALD AMN space. And then I think the final thing is, as a patient who prospects, kind of evangelizes the disease and the community to biotech companies, there seems to be a chasm between the big biotech firms that are interested, because we do have a well-organized community. We have a very hungry population that wants solutions, but we're still very small, and so they're hesitant to really commit to a program. And then we have the very small biotechs that are very excited, think they have a small molecule that could make a difference, but are short on funding. And so it seems like, I would say Minoryx is the ideal sweet spot where they have funding, they have the right size, and they also have the means to execute on a well-run clinical study.
How about some more questions from others in the room? Thank you, Ben. Those are great thoughts. Let me know about time too, so if we're getting close. But can I ask one? Question about data availability. What are the, I guess, evolving kind of parameters and sort of self-regulatory features of publishing or releasing patient data that is relevant to our understanding of basic disease? So just symptom progression, let's say in a placebo arm or even a negative result arm of a study. Can you comment on why a company might not release those data? Because I think for all of us sitting here, we say, well, of course you would release it. Why wouldn't you release it? But tell us why companies don't release that, or why we wouldn't, and convince us that that's the why that's necessary, I guess, in a sense. You can comment on whether it's the plan for your company to do it if you like or not, but maybe just as a kind of a broad sort of sense of things. Scott, just so the families in the room understand, understanding what happens to any of us in life, but particularly in this context, people with some form of ALD or AMN, is really important. I think this is the thing we all need, that the companies need in order to plan their studies. They need to know what to expect with and without treatment. And so this is really essential data. We call this data natural history data. And the term natural history is a term you may, if you're not familiar with, it's one you should become familiar with. What it means is what happens if we do nothing. This is really the data that matters greatly to certainly our community, but also to the companies themselves who are trying to plan trials. So maybe comment on why you would or would not release that kind of data from a trial.
Well, I think we have a responsibility to release data. On one hand, so when we planned the study, we benefited a lot from you. And we were able to look through Kennedy-Krieger data from Lorenzo's oil that helped us pick the primary endpoint of our trial. We are grateful to Mark Engelen, who gave us a look into his natural history cohort to see which parameters are sensitive to change and which not. So that means, on one hand, on the patient and the scientific community helped us a lot to plan our study. So I think we have a responsibility to pay that back one day. If the trial goes bad or it goes well, we don't know, but we should be able to release data. We're also discussing at the Industry Advisory Council, can we later on share non-sensitive placebo arm data, for example, for the benefit of everyone. So we should be doing that. So why are companies hesitant, or what happened in my own experience in the past? One thing is investors. You cannot announce negative data unless you have the go-ahead from investors. So it may be sensitive to the share price. If you're a publicly traded company, you have to release share price sensitive data within short time. But if you're a private company, it is not that strict. So as a scientist, I now also have to talk to the financial world, not only to the scientific world, and their thinking is different. The other thing is sometimes it's group dynamics in the company, that if you see a trial is negative, the whole study team falls apart. People are leaving the sinking boat. People lose their interest in working in the study anymore. So even if you have a good will to announce results, negative results in detail, sometimes you say, if I just place a sentence on clinicaltrials.gov, I've done my duties and I've publicly announced it. So it is also like a mental or psychological problem very often. It's just the team is evaporating.
I was just going to follow up. I mean, all of us up here are in companies that have some type of funding or we wouldn't be here, right? That is the challenge of running a biotech company and to keep the funding going. Fortunately for DoTareq, Poxel acquired our asset, but our program sat in phase 1 for over a year because we couldn't afford to keep it going and raising the money. And so, but if we weren't working on it, then as Uwe said, who's going to do the work if there's nobody left at the company? So it comes down to real practical matters. I mean, we continue to present posters. We have a draft publication or draft in progress of our phase 1 results. But I think all of us would agree it's in the best interest of the patient community as well as the regulatory and other companies to get the data out there. Sometimes it's just not practically feasible.
Yeah. Hello. We're very committed to transparency and to providing the data to the community as we obtain it. We have the benefit of open label studies. So we early on decided that annually we give updates. We update the community on the data. In the event of CALD with Lenti-D, the data has been positive, but we have had programs where early results were not as positive. Our sickle cell disease program is a good example. Our first patients treated in sickle cell disease with our lentiglobin product did not have the outcomes that we would have liked. However, we were fully transparent with those results, and actually, because of that, we're able to improve on the results. where our challenge is, so we do have a natural history study. We also have an observational allotransplant comparator study. And when we developed those, especially the allogeneic stem cell transplant comparator study, when we prepared that protocol and the consent, we tried to think ahead in terms of what could we include in the consent so that we would be able to share that data, that patient level data, with the community when appropriate. And we actually didn't get a whole lot of patient consent allowing us to do it. So that's been a challenge that we've had. So I'd love to brainstorm how to motivate more patients in these trials to actually give their consent for that broader data sharing, because that personally has been an obstacle for us.
Yes. Yeah, sorry, I couldn't help myself. So basically, as we discussed on numerous occasions, so patients are willing to donate their data if it helps drug development. So there is no question about it. So especially in such lethal or unpleasant diseases as we're discussing today. So the challenge is, and then, so when we're talking about clinical trials, basically any consent I've seen in clinical trials says that the data belong to the company, and company is, it can do whatever it wants with the data, including donating to the community. And there are efforts, Critical Path Institute, as you know, probably in Alzheimer's and Parkinson's, they're putting data together. We put data together for ALS and so on. So it's given. It's been done for the past probably 8-9 years. So in clinical research, observational studies, it's a little bit different, natural history studies and so on. But again, the clinical language and consent language is almost ubiquitous nowadays for data sharing. So I don't see any problems with that, and we're happy to share with you guys whatever we have. And it works in our experience in 12 diseases already.
I think part of what you're getting at, Alex, is it always, I mean, this is true of any kind of consent or agreement or dissent, is it depends on how the idea is proposed, right? So if they understand that, most of the patients understand that there's a benefit to the community or something like that. And, but I think the idea of sharing personal information may seem threatening if it's not put in the right context, to say this isn't for us to sort of use to market to you or something like that you might think of in like an Apple disclosure, for your iTunes agreement, but it is actually intended to be for a public benefit in some senses.
You're correct. And
I think
actually
it
in Europe, so there is a new regulation of data privacy or privacy, so GDPR, and one of the things they separate from everything else, from credit card information or Facebook information, they're saying if information is collected for clinical research purposes, this is for public benefit, and it should be treated separately and shareable. And anonymized data is not being considered non-shareable because it's by default, there is no way to get back to the patients.
Gary, your comment came as a surprise, and I'm wondering, did you all look into that to figure out why people were reluctant to engage in these consents, and really was it that portion of it? And did it cut across all Bluebird Bio products, or was it just in the ALD experience? Because I'm shocked by it, honestly.
The ALD experience was the only one that we had this observational comparator arm, so we didn't have the same situation in the other programs. We did not, I don't have an answer as to why that is. I think the way Alex just put it could be a good driver, is that, the way it was maybe positioned in the consent was not clear in whatever way. Maybe there's something that we could, I think he's right, that we could work on that language that could help improve this in the future.
I think Amber got a thought here too.
Just a thought. This is where you really can leverage the patient advocates, because I know, I'll speak bluntly, I've put out to other patient advocates that if you are enrolling in a trial and the company does not commit to making your, not the proprietary, the non-proprietary information public, do not consent for the trial. So I'd be happy to support those trials that are doing the right thing. So I can speak for ALD. You've got a whole room here today that would love to get you patients on your trial in the right way. So back to the points raised earlier, sometimes, as you know, consent forms can be huge. And this is just one more thing that they have to consent to, and it's easier to say no. So leverage the broader organization so we can all do the right thing.
I was also going to put Amber on the spot to share a bit more of actually the deep backstory behind gene therapy and its current form in ALD. The reason I wanted to share it is because it's, in a sense, this is a really crucial therapeutic idea that originated for our community within a member of our community to an extent, and I wanted to let other families hear that story, and it's a very inspiring one. I think you'll appreciate it.
So the story actually started in the year 2000 when our nephew was diagnosed with ALD, and at the time you could not fill a room like this with people who gave a hoot about ALD. So when we were looking for treatments, there was really not much out there. As we looked to see what opportunities there were to partner with academia, because obviously industry in those days were not doing anything in rare disease, except for Genzyme. I'm sure there's former Genzyme people, so thank you, we love you. There's Sanofi that took them over. So it was a struggle, as you can imagine. When we looked at what was out there, as we know, we still don't fully understand all the pathophysiology of it. So when we looked at gene therapy, it was this very, in some ways, attractive thought that, well, we know it's a monogenic disease, or at least that's the belief, so let's just fix the gene, and then we don't have to worry about how it really works. We'll just fix the gene. When we started to do research into who was doing anything that we could leverage, and part of this had to do, I simplify the story. I was very fortunate that I was an executive at a major pharmaceutical and worked for the head of R&D and said, what would you do if this was your kid? He's like, do gene therapy. And having worked in pharma at the time for, 15, 20 years, I'd never heard of gene therapy. So he's like, talk to my friend Jim Wilson, he'll tell you all about it. And mind you, this was right after Jesse Gelsinger had died on a gene therapy trial. So when I started talking to my GSK colleagues about doing gene therapy, they looked at me like I was completely insane. But I said, well, there's no other really ripe opportunity to go by. So Rachel and myself spent some time looking at who was doing what out there, and that's how we ended up chancing upon that there was a small program in France under the leadership of Patrick Auberg, where he was so frustrated not being able to do anything for this disease, and even though he was a neurologist, he's like, all right, gene therapy, he had the same approach. We know that we can fix the gene, maybe that's the way to go about it. So we basically spent quite a bit of time in a cold, pretty miserable conference room, actually talking through for about 4 or 5 hours what a gene therapy program would look like. And back in those days, no vector had actually been in humans to do an ex vivo approach like this, and I won't get into the details of why we decided to do ex vivo lenti versus, in those days people were doing retroviruses and such. But the net of it was we really had to team together, figure out how could we potentially get a vector which cost millions and the people involved to get such a trial forward. A lot of it had to do with, we'll put aside that we got companies involved to move forward the use of their vector. And in those days it was Cell Genesis, because Cell Genesis wanted to use a lentiviral vector for other diseases. And the way we tried to convince them to use it for ALD, I'll never forget the phone call to Steve Sherwin was, boy, do I have a deal for you. I can get your vector into a human way faster than any other disease because I'm working on a disease that's killing kids. So there's a less of a bar, back to the points earlier in terms of what kind of threshold would people be willing to risk in terms of that risk benefit. So that was really what led to Cell Genesis making their vector and people available. And there were some people who got involved, not just because they were asked to do so by Cell Genesis, but because of their personal passion. And I know you guys still work with Orpheris at Bluebird. I mean, he was one of those at Cell Genesis that even as that company went through changes, he felt personally passionate about driving that forward. But a lot of it really came down to the entire community, the entire infrastructure just pushing against it. I can tell you, we both attended RAC meetings back then when the risk-benefit was questioned. How could you go into humans with this? And we really had to paint the picture at how bleak it was for these kids, and that they felt that it was a risk worth taking when you look at the alternatives in terms of this horrific disease. I mean, it's fortunately so different in the gene therapy space since then, now that there's thousands of people who have been exposed to vectors. There's been a lot of trials. I won't say there's been a huge amount of successes, unfortunately, but we know that they're safe and well tolerated. Now the question is, can we get enough of the protein expressed in the right cells? But I will say the net of it was just that perseverance and patience, going to the right forums, going to the right places to make sure that we got the resources, that we got the buy-in, because no regulator wanted to be the person who let thalidomide go through in the US. And you really had to convince them what the risk benefits were and what the case was. And be responsible and hear what everyone had to say. And as I said, even when we were in human trials, my GSK colleagues looked at me like, it'll never work, you're crazy. And I will say personally, and I'm really curious what Rachel's views, I don't know if I ever said this, I did not think it would work in the first 4 kids. I thought we would just learn from that what we would have to do for the next. Because in those days, we didn't know how many cells needed to be corrected. I mean, that was the big question. I kept saying, well, heterozygotes do fairly well with 50-50. I mean, they're not dying of cerebral for the most part, but we really didn't know. And as you know, and I'm looking at Bluebird people, we're not near 50%, but yet it's good enough. So we really have to be bold and aspire and think big picture and not let any of the obstacles or people stop us. Respectfully hear what they have to say, because even when people were disagreeing with us, there was usually a nub of an idea there that we had to take on board and really consider. I mean, as much as you always have to take that balanced perspective with the safety risk and what the implications are for completely killing the gene therapy field beyond where it was at the time, or what would happen in terms of other patients and trials. You never want to do something that will set things back even further, even with all good intentions. But I'm curious, I'll pass it on.
I think Amber pretty much told the story. I think what helped me, I guess we've really never talked about this, what gave me some ballast to think it would work is that right when we entered the field, a very important stem cell transplant paper had been published that showed that allogeneic stem cell transplants can be effective in cerebral ALD. I'm sure Troy's familiar with all those data. And that, to me, kind of was the gold standard. And Amber would always remind me, the benefit of gene therapy is patients are going to get back their own cells, so we're not going to have to address all of those graft versus host, which can be so lethal or debilitating. So I guess in this situation, because there was a gold standard, I had some reason to be more optimistic. And also, even though Amber went through a lot of the challenges, she still hasn't scratched the surface because so many other things were happening with other gene therapies that at one point, understandably, the sponsor had decided to shut the trial down, and we had to go into hyper-Salzman mode to get it reactivated. And if any of you have ever come across hyper-Salzman mode, it's very effective.
I can tell you that Jim Wilson said that he was afraid I would follow him into the men's room. I didn't, but I came close.
The thread there. So Amber's son Spencer was affected by ALD, and I think it's interesting to frame it in this light where we think that a single individual actually, in this case really Spencer, inspired something that is now flowing into the lives of many families and children. It's a very, I think, inspiring story. And I think the other part I wanted to illustrate was the crucial role that family members, now granted, very savvy family members already in the industry, so to speak, but were key to navigating and persisting and making that happen. So if not for Spencer and Amber, Bluebird Bio would not really be working in the ALD space. So I think it's important for us to sort of see these connections as they play out over time, and to recognize that that this is how the sort of organic and often very slow-moving way in which these things happen. But this is real progress. So I think we're at close, but I think it's worth also taking into perspective the 5-year time capsule since we started ALD Connect. It was in 2013, our first meeting. I think at the time either Bluebird was just opening or had not yet opened its first study, and I'll just maybe gloss over that detail by saying there were essentially no trials running for ALD, and there really were very few in the wings. And I think here we are, 5 years later, and we've already got several trials running, and several trials we hope will be running soon. It's it's really leaps and bounds of progress for our community. So I think a big round of applause to the companies and to the families and to the patients. So.